So this lecture is going to be about what the nose knows, so the olfactory system, and how simple odorant becomes an odor, and why it's such a powerful part of our memories and part of our experience in the world, and how that chemical um, component of an odor becomes a smell in our brain. So first off, a couple of facts here. Every species uses a sense of olfaction or chemosensation, except one, and that's the cetaceans, which are these cute little dolphins right here. Those dolphins do not have a sense of smell, but pretty much everything else has a way of chemosensation, whether it be smelling or recognizing a chemical and being attracted to that chemical. Um, so for humans, we have about 10 square centimeters of olfactory epithelium. Okay, not very much, especially compared to some dogs who have up to 170 square centimeters of olfactory epithelium. And the olfactory epithelium, that's the place where the receptors are located. They're going to respond to our cells, and we'll talk more smells and different odors. We'll talk more about that later. A dog's olfactory epithelium is also way more densely innervated, okay, up to about 100 times more receptors per square centimeter too, versus humans. So obviously their sense of smell is much more important to them than it is to us. It is a very important sense for us, but it is not as powerful as it is for, say, a dog or lots of other mammalian species. Anosmia is the loss of a sense of smell. So someone who is an anosmatic is someone who cannot smell anything at all. Maybe a nerve's been cut, the olfactory feeling's been damaged to the point where they can't smell anymore, or something's happened where they can't smell anything around them. Okay, so how does an odor become a smell? So first off, we have a couple of things. We have odorants that are floating around. They're volatile compounds. They're going to bind to what are called olfactory receptor neurons, which are in the olfactory epithelium. They have G protein coupled receptors that convert that chemical to an electrical signal. Then we have these olfactory receptor neurons are going to project to specific what are called glomeruli in the olfactory bulb. So the olfactory epithelium is separate from the olfactory bulb. Okay, it's associated with it, but it's separate. And then these mitral cells that make up the olfactory receptor, these glomeruli, okay, are going to take that signal from the bulb and then send it off to piriform cortex for further processing. Okay. Notice the one step that's missing here, as opposed to taste, is that we are not going to the thalamus. We're going straight to this thing called the piriform cortex, which is one of the oldest parts of cortex, because the sense of smell has been around. It's one of the longest standing senses that's out there. So a little bit about nose anatomy. Okay, main things here. We have a thing called the cribriform plate, which sits as a small bone in between the olfactory epithelium and the olfactory bulb. And the olfactory bulb, again, remember from our anatomy, sits kind of at the base of the brain. So inhaled air will come up, it'll go into that olfactory epithelium, bind to these olfactory receptor neurons, which we can see here, the olfactory receptor cells here. It's going to bind to the cilia, which is stuck in the mucus layer. The mucus layer is what's going to help catch those odorants, catch those volatile compounds. So they actually bind to the cilia of the olfactory cells. You can also see we have these supporting cells here, which provide kind of the structure for the olfactory epithelium. And then we also have these basal cells here. They're really important because that's where we can actually generate new olfactory receptor neurons. They're going to be replaced on a pretty consistent basis. And the interesting thing here is that these basal cells are going to exactly mimic the olfactory receptor cell that they replace. Okay. So you zoomed in here again, we have that cribriform plate, a little kind of looks like a grater, basically, with tiny little holes in it for the axons from the olfactory receptor neurons to pass through, create those olfactory nerves, and go into the olfactory bulb. So what happens to that olfactory receptor neuron? Okay, again, here's your cilia. We zoom in on that cilia right here, and then we have these olfactory receptor proteins. They're the G-olf proteins, okay, the golf proteins. They are going to basically react to an odorant molecule binding that odorant receptor, and then that's going to activate an adenyl cyclase, which activates a cyclic AMP pathway, which allows calcium and sodium to come into the cell, depolarizing the cell. And to further depolarize the cell, chloride is actually ejected out. And all of this leads to depolarization, which creates the action potential, which goes down through the olfactory receptor neuron up to the olfactory bulb. So that's our kind of chemical process of how the action potential is generated, how those potentials are generated. But here's a kind of zoom in on the electrical the potential generation. In the cilia, you just have the receptor potential, so you get this strong depolarization. Then when you get to the olfactory receptor cell itself, then you start to generate action potentials, okay? And then the olfactory nerve transmits those action potentials out to the olfactory bulb. So you're going from a receptor potential, no action potentials at the dendrites, to generating those action potentials in the soma and out the axon. So a couple things here. Each neuron expresses exactly only one olfactory receptor gene. So it only expresses the one receptor per olfactory, each olfactory receptor neuron only expresses one gene, one specific. There are about 1,000 different odorant receptor genes in humans. It actually makes up about 3% of your genome, which is a lot. So you can have each one of those different types of like about 1,000 different olfactory receptor neurons, each one only expressing, again, one receptor gene. Now, it doesn't mean you have 1,000 olfactory receptor neurons. You have a lot more than that, but they each only express one of those 1,000 different possible genes. So what does that mean? Well, it means each receptor can only respond to a limited range of stimuli, and they usually have a dose-dependent response. So again, dose-dependent meaning how high of a concentration is there of the odorant that comes and binds to it. Again, as I talked about earlier a little bit, these neurons can be regenerated. Those olfactory receptor neurons are one of the few areas in the brain where neuroregeneration takes place on a regular basis. Those basal cells will become the olfactory receptor neuron that's closest to it, and they will basically do a one-for-one exchange with expressing the exact same receptor neuron, accepting receptor gene, sorry. So it's kind of a trade-off of just every couple of weeks or so you flush out and you get a new olfactory receptor neuron, but it's one that exactly replaces the one that was previously there. So all this was found out by Linda Buck and Richard Axel. They received the Nobel Prize for identifying that gene family for olfactory receptor neurons. So how does this kind of work? How's it laid out in the olfactory epithelium? We can see here we might have a bunch of different neurons that all express the same olfactory receptor gene, but we can how can we use that to create a sensation of expressing tens of thousands of different odors, right? We might only have 1,000 different receptors, but we can really sense a lot of different odors and odor combinations. So how this works is kind of like with taste, we have this thing called population coding. So you have different ranges of affinities for receptors. Some cells are going to be more sensitive than others to some odorants, and concentration is going to matter. So if you look over here on the left, you can see, like for citrus, for example, it activates the green cell a lot and activates the blue cells a little bit, and it doesn't really do anything to the red cells. Okay, so that combination of green, blue, and red would indicate to the brain that that's citrus. Okay. Floral looks like it does the green cells just a little bit, the blue cells a lot, and even gets a couple little action potentials on the red cells. So what does that tell us? Well, that's the combination that's going to be a floral scent. So the message is coded in the levels of activity across a whole population of neurons. So even though they only express one receptor protein, they will still respond to multiple odorants, but to different levels. And we can kind of see that on this next diagram here, where they had 25 different cells, and they looked at the response to different odorants to each of these 25, to different odorants at each of these 25 neurons. And they saw that, say, for N-amyl acetate, for instance, 3 different cells responded to it, okay, but to varying levels. And you can see cell 3 actually responded to N-amyl acetate and cineol simultaneously, or not, responded to both of those two odorants, but to different extents. So what is this? Or you look at cell 17, especially, responded to benzaldehyde, cineol, and 3-heptanone. But it responded to each one in a different way. So what does that tell you? Well, it tells you that these neurons aren't specific to a given odorant, but they are going to respond to different odorants in a different way. So that's kind of important for determining population coding. So now we got through the olfactory receptor neurons. Okay, we want to know what happens after that. So in the olfactory bulb, you have these things that are called glomeruli. The glomeruli are basically the place where the olfactory nerves from the olfactory receptor neurons join up, those axons join up with the dendrites of what are called the mitral cells. The mitral cells are these red ones here. They're the ones that are going to send their dendrites out into this glomerulus. So the glomerulus doesn't really have cells in it. It's more just a combination of axons and dendrites from different cells converging. So you have about 25 different, see there in the stats, 25,000 primary olfactory axons, so coming from the olfactory receptor neurons, are converge on about 100 second order olfactory neurons. So you don't have as many of the olfactory neurons, second order neurons, as you do of those primary receptor neurons. And one of the things that's cool here is that cells expressing the same receptor that are distributed throughout the olfactory epithelium are going to tend to combine at the same olfactory, the same glomerulus. Okay? So this helps create like a spatial map almost of odors. So, sorry. So you can see here, like the green cells, they all combine back to one glomerulus there. The red cells are all going to send their receptor neurons, even though they come from different parts of the olfactory epithelium, all the same glomerulus. Now, it doesn't mean a specific glomerulus only responds to that receptor. No, it doesn't. We don't have that many glomeruli for that to work, but all of the ones that do express the same gene, receptor neurons that express the same gene will converge back onto that single glomerulus. And you see that here. Basically, they did a GFP labeling of the glomerulus from one single olfactory receptor neuron receptor cell, and they tracked the dendrites where they all, they tracked the axons where they all converge back onto, and they saw this beautiful like single glomerulus layout in the olfactory bulb of the mouse. The other thing that's pretty impressive too is the symmetry. So these two glomeruli are basically on opposite bulbs, but you can see they're basically at the same location on each bulb. And it would actually be the same across multiple animals and be almost in the exact same place. So a lot of this is established in development. And it maintains that symmetry, like I said, across species. So that glomerulus would probably look the exact same or really close to the same on even another animal that you looked at. So one thing we saw here is the glomeruli have moderate specificity, but they're not extremely specific. So they looked at different glomeruli. Up above, they did some calcium imaging, and they just kind of looked at how active the cells were based on that through a calcium dye, indicating how much calcium is coming into those dendrites right there, into those glomeruli. And you can see for all these different odorants, okay, EG, MEG, IEG, whatever, it doesn't really matter what they are. They're all just different odorants. Different glomeruli were active to different levels, but they weren't specific to a single odorant. Okay, so glomeruli have a more moderate specificity than, say, the olfactory receptor neurons do. Okay, you can see they're more active to lots of different odorants. Doesn't mean that the same axons that are coming in are active in each case. It just means that they aren't as specific as other parts of the, as the further out, more distal parts of the system. Okay, so cool. So now we got that. Where does everything go from there? Well, so we have our olfactory receptor cells, we have our olfactory bulb, okay, and now we have our projections. And some of the projections are going to go back to what's called the olfactory tubercle, okay, and that's what's going to go to the thalamus, and that actually goes to orbitofrontal cortex, okay, which is where you have your decision making. So some of your sense of smell, okay, goes straight in, does go through the thalamus and into cortex to help make you process, kind of help process your decision making, your interpretation of what's going on. However, most of the information will actually bypass that tubercle and go straight into olfactory cortex in the temporal lobe. And that's called the piriform cortex. Basically, that's the second stage of the signal interpretation. That's where the actual processing of this combination of glomeruli being active to this level is this smell. So that's the kind of the major pathway for the olfactory system. The secondary pathway is the one that's going through the thalamus to the cortex, which is not saying this is what the smell is. It's saying more this is my reaction and this is how it's going to affect my planning based on seeing this smell. And again, the rest of the brain is going to, the olfactory cortex is going to process that information too and send it out to everywhere else. But we are going to bypass that primary information. We are going to bypass the thalamus for that primary information and go straight to cortex in this system. So one other part of the olfactory system that is around in most mammals is the vomeronasal organ. That's what responds to those things called pheromones. Nobody's really found the vomeronasal organ in humans, okay? But in lots of animals, it sends out really important signals. It sends signals about reproductive behavior, territorial boundaries, kind of identification. It can lead to aggression. This is why like dogs go around and sniff other dogs' butts. That's where they get those pheromone signals to kind of identify what species, what territory are you ready for reproduction, all of that thing, kind of getting identification off that. Mice use it too. There's vomeronasal organs kind of in the anus area, but there's also some in the eyes, like near the eyes. So mice will like sniff each other in the eyes too when they get around each other. And in that process, they're taking the pheromones. And it actually shows that you can mess with reproductive behavior and cause gender confusion and odd mating patterns in mice by manipulating the vomeronasal organs or removing them, or removing like which pheromone or manipulating which pheromone the mouse gives off. You can actually cause all sorts of chaos with the mice based on kind of their perception of what that animal should be. So kind of wrap up chemical sensation. So we've done taste, we've done smell, and getting that process of how do you take a chemical from the environment and turn it into a perception of what's going on. So we got to the initial part of how do we take the chemical, make an electrical signal, pass it on, what are the mechanisms early on, and then how does that end up becoming, and then the brain ends up processing that information based on the coding. And that's one of the big things is the population coding for both of those things. And the fact that there are basically these sensory maps, right? The glomeruli are mapped out in a way where, you know, a certain pattern of them to a certain extent in each way is going to generate each of the different smells that we've perceived throughout our life. And the brain will recognize that pattern and turn it into, hey, this is an orange, or hey, this is a banana, or hey, this is a lemon, as opposed to perceiving the individual tastes, the individual odors themselves. You perceive the whole population together. And a lot of it will come down to what they think is timing of action potentials. So it's not, for population coding, it's not just about how many are active, but it's about when they're active too. So it's probably going to be that timing aspect is going to be really huge in terms of understanding, okay, this one's active now, this cell A is active to a certain extent, then cell B, then cell C. How is that going to play out in terms of what I recognize from past experience? So hopefully you have a pretty good understanding, at least of the basics of how we perceive the chemical world around us. And then next we're going to get into vision and how we perceive the optical world around us.